As my mentor just said, the team and I are very excited to have you all here this morning. So with that being said, let's get started. Raise your hand if you see something on the screen that you do every day of your life. So whether it be walking your dog, walking the halls, or sports. Yes, that's all of you. So keep that in mind for the topic of my paper. The topic of the paper that I focused on is hypertrophic cardiomyopathy, or for short, HCM. I will be referring to it as HCM during the presentation. It is an inheritable genetic disease caused by the MYBPC3 gene. Now, what that gene does, it actually is it is responsible for 40% of all of the genetic defects that causes HCM. And what it does, it encodes for a signaling node in your cardiac myocytes. Now, these are just the heart cells that make it beat. So they are what actually contract and relax in your heart. And what happens in the face of this defect is they get bigger. Now, what that does, it actually causes your heart itself to get bigger. Here you see the myocardium, which is just the muscle wall that separates the two atria. It's swelling a lot. And what that happens after that, you actually get functional problems. Here, displayed by arrows is actually blood flow. So you see it entering the heart via the superior vena cava, and then as it tries to exit out of the left atria, you actually get regurgitation back down into the left atrium. So that can cause a lot of symptomatic problems like arrhythmias, palpitations, and even heart failure. HCM is actually the leading cause of sudden death in young athletes. So just to give you an idea of how common this is, it actually does have an extremely high frequency, 2 to 8% in Indian countries, which is about 1 in every 500 adults, and that's just adults. Just to give you an idea, how many of you have heard of BRCA 1 and BRCA 2? Yes. The prevalence of BRCA 1 and BRCA 2 is only 2% worldwide. And it's not just physical. Remember when everybody in here raised their hands seeing an activity that they did every single day of their lives? Could you say that you wouldn't be scared of doing those things, like walking your dog or going for a jog, if you thought that you could die from just doing those simple things? So it is a very, very psychological disease as well. So some of the current treatments of HCM are beta blockers. Now these are just pills that help to thin the blood so the regurgitation doesn't cause a lot or as much as swelling. My uncle actually suffers from a form of cardiomyopathy. It's not HCM, but he does take these beta blockers and he has to take them every single day of his life. Another current treatment is surgery. So this includes a myectomy, which is basically just cutting off some of that myocardium to make space and reduce regurgitation, and ablations, which is just kind of trying to break up the blood via alcohol by a catheter into the heart. Another one is heart transplant. Now, many of you know that transplant lists are very, very long, so most patients actually die waiting for their new heart. Now, another treatment regarding people who are trying to have babies who maybe carry the mutation for HCM is IVF. Now this just stands for in vitro fertilization. The only problem with this is that it is very, very expensive, running patients anywhere from $12,000 to $20,000. Now mind you, this disease is genetic, and so it doesn't discriminate against class or against means. And most of the people who actually have this defect don't have the money or the means to pay for IVF. Also, all of these treatments are offer purely symptomatic relief. It's not attacking the root of the problem, which is the gene. So, this is where my paper comes in. Correction of a pathogenic gene mutation in human embryos. Pathogenic just means disease-causing. So, the superstar of the paper was CRISPR-Cas9. Now, this is a gene editing tool that goes into the cell, the nucleus of the cell, and actually breaks apart your DNA via a double-stranded break. That's what DSP stands for, to either edit in or edit out genetic information. Now, it's composed of 3 main components. Here, pictured in blue, is the Cas9 protein. Now, these are the scissors of the whole tool. This is what actually is inducing the double-stranded breaks in your DNA. Then you have your guide RNA. Now you can kind of infer what this guide RNA does. It guides this whole tool to the desired region of the DNA. Then you have your tracer RNA down here. Now this is what actually latches on to the double helix of the DNA in order for CRISPR-Cas9 to do its job. Now what's really cool about CRISPR-Cas9 is that it's patient specific. It can work in elderly people, it can work in young people, men and women. It is very inclusive, but just keep in mind that this sort of treatment would be more geared for adults trying to have babies, and they can detect HCM in the embryos. And also, this is a long-term solution. So if you think about it, over the next several generations, if we use this as a tool of editing HCM out of the gene, the future generations to come wouldn't have to suffer from this genetic disease. So it is a very, very long-term solution. Any questions so far about HCM or CRISPR-Cas9? Yes?
When they use the surgery to like shear off a part of the middle muscle wall,
Yes.
does it grow back? Like, do you need multiple surgeries, or do you know how doctors usually handle that?
That is a very, very good question, and I don't know the definitive answer, but my educated guess would be yes, the tissue and the muscle could run the risk of building back up, just kind of like when you get your tonsils removed. Some people actually grow them back. This is just how your body responds to damage, tissue damage, yes.
How do they detect it in embryos?
In embryos, okay, that's a very good idea. They can run an amniocentesis sometimes to see if those cells carry the HCM gene. It's also, if you run analyses on a cell, it actually shows up. It's a GGAGT mutation, which is in the NYBPC3 gene, so that's how they would detect it. Yes.
You had mentioned IVF as being treatment. Is
Yeah.
that kind of more what they're going for here with CRISPR-Cas9 kind of future generations?
Yes, correct. Any more questions before we move on? Okay, so there are a few ways that your cell can respond in face of CRISPR-Cas9. So the first one is NHEJ, which is over to the left in that picture. Now that stands for non-homologous end joining. So what your DNA is actually doing, it's compensating for that lack of information or gap in your DNA with an indel. Now indel can either mean a complete lack of DNA, so they would just kind of glue those two ends of the DNA together, or they put random base pairs that have no meaning in just to kind of use as a filler. So the next way that your cell can respond is via HDR or homology-directed repair. Now it's the repair of the double-stranded breaks with a donor DNA. Now this can actually come either from the cell or it can be exogenous, meaning that is introduced to the cell. Now in terms of this paper, the scientists introduced a synthetic form of DNA, and they just call it SSODNs, which stands for oligodeoxynucleotide. All you need to know is that it was made by the scientists. Any questions about that? So the first thing that they did was they derived and cultured iPSCs, which stands for induced pluripotent stem cells, from skin fibroblasts from the patient. Now iPSCs are just basically taking a cell that has a lot of information to make it specific and stripping it down of all of those markers and all of those all that information to make it kind of a blank slate cell. Does that make sense? Okay, so what they did was they transfected these cells with nucleofection and electroporation to introduce the CRISPR-Cas9 complex into the nucleus. So they just poked holes into the nucleus and then injected the enzyme, the SSODNs, and the guide RNA. And then after that, they propagated these cells, they cloned them via cell division. Does everybody understand? Okay. So what happened? Here on the y-axis of the graph all the way to the left, you have percent HDR. Now remember that that means homology-directed repair. So that's how the iPSC actually repaired the lack of information in the DNA. And so over here on the y-axis of this graph, you have percent NHEJ. Now that means, again, non-homologous end joining. And you can see that the CRISPR-Cas9 complex in the iPSCs we're actually using a lot more, we're going the route of NHEJ a lot more than they did HDR. So in comparison, iPSCs use NHEJ more of the time than they use HDR. So now let's just zoom out for a second because we're going to move on to a paper that doesn't really have a lot to do with these iPSCs, but you'll see how it connects in the end. So then they wanted to see, okay, iPSCs are really great for I guess using it as a method of gene correction, but embryonic stem cells are the strongest. They're the superstar of gene correction via CRISPR-Cas9. So what they did is they took M2 oocytes. Oocytes are just eggs that are used in fertilization and reproduction, and it was in the meiosis II stage of the cell cycle. And what they did was that they actually co-injected the mutant sperm, which is over here, that actually has the mutant MYBPC3 gene, and they washed it with the CRISPR-Cas9 suspension. And then they injected that into the cell and let it divide and develop. Any questions about that? Okay, moving on. So how did the M2 oocytes respond? Now over here to the left, you can actually see the targeting outcomes and part of the genotyping of the results. So over here in blue, pictured in blue, is wild type. Now that just means these cells were normal and there were no detections of the HCM mutation. And pictured here in brown is percentage of the wild types that use NHEJ versus HDR in repair. So now we're going to talk about HDR. So this is with and without that synthetic DNA that was introduced by these scientists. So over here you have the plus SSODNs, and these are the percentage of oocytes that used this, this given DNA in its repair route. Over here, this is where it totally ignored the not fake DNA, but synthetic DNA, and used something else. Now, does anyone have an idea of what it could possibly have been using? So think about it. Remember, HDR could either use information that was already in the cell versus information that was introduced. So what it bless you.
Just the information from the cell.
Yes, but if you think about it, maybe what allele? Okay, so yes.
Is it from the non-mutated allele of the
Yes,
mutated thing?
which was the maternal allele. So it's actually completely ignoring the I guess synthetic DNA and using what was already there. So let's take another step back. The scientists also wanted to see which part of the cell stage was most efficient in yielding the best type, the best embryos. So what they did was they took an S-phase zygote, and instead of co-injecting the CRISPR-Cas9 and the mutated sperm, they fertilized the egg with the mutated sperm, and then they waited 18 hours, and then they introduced the CRISPR-Cas9 complex. And over here, you can see that there was a number of different genotypes, which I'm going to get into later, but that's definitely something important to remember during the rest of the presentation. So how did the S-phase embryos respond? So over here, all the way to the left, you can see that compared to the control embryos, which were half and half mutant and wild type, the S-phase embryos were corrected a lot. You can see pictured in the blue that a lot of normal genotype cells with the normal genotype were represented. But you can see that there's the mutant genotype, but there's also something else, the mosaic genotype. And that basically means that within the actual zygote, there were a number of different genotypes represented. And this is something that the scientists did not want when thinking about clinical applications. So also targeting outcomes in mosaic embryos. So here you can see at the bottom is the with synthetic DNA, with and without synthetic DNA. And you can see that it didn't really affect the mosaicism of the cell, which means, again, multiple genotypes of the embryos. So it didn't really have a big impact. Now to take another step, let's just look at the overall targeting of CRISPR to see if it's actually doing what it's meant to do. So you have overall targeting efficiency in the embryo. embryos, and the majority of that, almost 3/4 of the embryos were targeted in the place that they were supposed to, and there was cleavage of the DNA at the desired location. Now, overall HDR efficiencies, as we talked about before, the zygote and the oocyte both preferred HDR rather than HEJ. So let's compare the two. So now the scientists moved on to post-isolation and analysis, so more repair talk. We have the S-phase injected embryos in the middle graph right there, and then you have the M-phase injected embryos. Now, what's the big difference? As I mentioned before, just to recap, can somebody answer that for me? Josie?
In the S phase one, they're like, do you mean the difference in the outcome or the actual application?
In the outcome.
Oh, well, there were like a lot of different genotypes in the S phase.
Correct. So you have that green bar up top, which represents mosaicism, as Josie just put. So based off of that, which of the two embryos, the oocytes or the zygotes, the S-phase zygotes, had more of a clinical application based off of the data I just presented? Would it be the zygote or the oocyte if the scientists don't want mosaicism? You back there.
The oocyte?
Yes, the oocyte. Very good. And so what then they did is they evaluated the cell integrity of the oocytes. Here you can see these 8-cell embryos. The morphology is normal. You don't have any big growths anywhere. And then the development into a blastocyst also is very normal. So just to recap again, the zygotes had mosaicism, which is what we do not want, and the oocytes did not. It resulted in a uniform embryo with wild type cells. Yes?
What does the mosaicism cause that's they don't want that?
So think about autoimmune disease. If a part of your body, a part of your cells aren't compatible, and some of it is, it can cause your body to attack itself. Any other questions about mosaicism? I know it's a little bit technical. Okay. So, now let's compare the iPSCs that we talked about in the beginning and the embryos. The ISPs, remember, they preferred NHEJ as a repair method, while the embryos preferred NHEJ. Here you can see below, it's just pictured percent HDR. The iPSCs, only not even half of them used HDR. Meanwhile, they just used S-phase embryos as the component of comparison, and they used, the majority of their cells used HDR. So here's just a schematic for you. Up here you have your patient embryos, and you can see that red marker on the second double helix that represents the paternal allele, which was the mutant sperm. And they didn't use, here pictured in blue would be the SSODNs, and they didn't use that in trying to fix the lack of information in the cell, while the iPSCs did use that SSODN in creating the complete DNA structure. Does everybody understand that so far? Okay. So again, just to give a brief summary, there are two possible clinical applications of this CRISPR-Cas9 in trying to fix HDM. You have your iPSCs and you have your embryos. Now, as I don't know your name, but as that gentleman back there had so well put, the cell integrity of the M2 oocytes was definitely better than the zygotes. But they also had different routes of repair. The iPSCs went with NHEJ, while the embryos went with HDR. But why? And that is what brings me to my open-ended. The scientists actually have no idea what the driving determinants of repair is in the early stages of development. So this is a whole other route that we can pursue in answering more questions to make this more safe in the future. And also the detection of mosaicism. That's also a big problem because it can cause a lot of autoimmune problems, and it's a big risk of CRISPR. And also just the ethics of it all. A lot of people believe that gene editing is not at all ethical and we should not be doing it. But in the face of these genetic diseases, what other choices do we really have? Does anyone have anything to add?
I actually have a question. So
Yeah.
I know you said they edited the sperm
Mhm.
sheets. Did they ever think about editing the egg and not the sperm instead? And like so they would use the paternal instead of the maternal.
No, they didn't actually do that because I did mention before, HGM sometimes is sex specific, so it's usually on the side of the male
Okay.
who carry the mutation. Anybody else have any questions either? Yes.
You said that this treatment would be used instead of IVF, so what's the price point difference?
So the price point wasn't at all evaluated in the paper, but that's a very good question. It is one I have myself, and I'm ill-equipped to answer that at the time, but I can definitely go into that and get back to you. Any other questions about anything? Well, thank you for coming. I just want to thank Dr. Kirkhart, Ms. Mingus, my co-head Matt, Jessie, who couldn't be here this morning, but she suffered through a lot of practice presentations. The team, Mr. D, who I guess nurtured my love for science. Amanda Celli, who was the previous head, she helped me a lot. My friends, my family, and all of you guys who listen to me talk about random science.